In computational biology, we constantly ask, are these two things moving together? Genes, proteins, metabolites, do they rise and fall in sync? Each correlation method is a different judge watching the same dance. Welcome to the dance floor. Pearson only sees straight lines. Watch these two dancers move in perfect lockstep along a diagonal. Every step one takes to the right, the other takes proportionally upward. Pearson gives a near-perfect score r equals 0.95. Now the dancers shift to a curved waltz moving together along an arc. They're clearly coordinated, but Pearson's score drops to nearly 0. The straight line judge is blind to curves. And watch what happens when one dancer leaps to the far corner, a single outlier. The best fit line lurches. The score collapses. One rogue footprint ruins the whole assessment. In gene co-expression studies, Pearson is the default in ANA. It works brilliantly when genes rise and fall together linearly. But it's blind to anything else. Spearman doesn't care about exact distances, only order. First, second, third. An assistant labels each footprint by rank. Now watch, the dancers trace a logarithmic curve. It's monotonic, always rising, but not a straight line. After ranking, this curve straightens into a near-perfect diagonal. Spearman catches what Pearson missed. And that outlier from before. After ranking, it's just ranked 20 out of 20. The wildest point gets the highest rank, but it doesn't distort distances. The score barely changes. In biology, use Spearman when your data is skewed, RNA-seq counts, protein abundances, anything where the row values aren't normally distributed but the ordering is meaningful. Kendall zooms in further. Instead of ranking the whole dance, Kendall looks at every possible pair of steps and asks, did both dancers move the same way? Concordant pairs both went up, score a green check. Discordant pairs 1 up, 1 down, get a red X, the ratio gives you tau. With small datasets, 10 or fewer observations, Kendall is more reliable than Spearman because it makes fewer assumptions about the overall shape. In proteomics experiments with few replicates, Kendall gives you a more honest answer. Two dancers look perfectly synchronized, but are they following each other, or are they both just following the beat? Here's a third figure on the floor, the teat. When the beat speeds up, both dancers move faster. It looks like they're correlated, but really they're both responding to the same external signal. Partial correlation puts on noise-cancelling headphones. It removes the dis influence and asks, with the beat factored out, are the dancers still coordinated? In gene networks, this is the difference between direct interaction and indirect association. Gene A and gene B both correlate with gene C, making A and B look correlated. Partial correlation strips away C and reveals whether A and B truly interact. 4 judges, 4 perspectives. Pearson for straight line sync. Spearman when the data is messy but monotonic. Kendall when you have few observations and need robustness. And partial correlation when you need to know who's actually leading versus who's just following the beat. Next time, judges that catch curves, rings and chaos.